Discuss about DNA in unit 1. Today we are focusing on DNA, right? DNA is deoxyribose nucleic acid, deoxyribose nucleic acids, right? And DNA, it's a deoxyribose nucleic acids, and in simpler word we can say that it's a polymer of nucleotides, nucleotide. What is nucleotides and what is the composition of nucleotide? What actually it is? So nucleotide is made up of nitrogenous bases. There is a sugar, there is a phosphate as well. So one by one we are going to discuss about what is nucleotides and what is DNA, how it makes the polymer of nucleotides, how they interact with each others, right? So DNA contains nitrogenous bases. We talk about nucleotide, right? So nucleotide contains nitrogenous bases, and another thing which nucleotide contains, it contains sugar and it contains phosphate, right? So these are the three substances which contains the nucleotide. तो nucleotide में तीन चीज़ें हैं, एक है nitrogenous bases, एक है sugar और एक है phosphate molecules. So one by one we are going to discuss. First is nitrogenous bases are of two types, purine and pyrimidine. Purine having the two rings, that is a purine. Single ring structures we call pyrimidine, right? So in purine there is a two bases, adenine and guanine. And in pyrimidine there is a cytosine and thymine. So word here you have to be give the attentions, thymine, THY you have to write. Otherwise if you are writing thymine, THIAMINE, तो thymine का मतलब होता है vitamin. So it's a vitamin, right? So you have to write THYMINE, thymine. So these are the nitrogenous bases in the DNA, in purine, adenine, guanine, and in pyrimidine, cytosine and thymine. So if we talk about RNA, RNA is a ribonucleic acids. So instead of thymine, RNA will contains uracil. So in nitrogenous bases, in pyrimidine, rest will be same. RNA की अगर compositions करनी है तो nitrogenous bases which are required in purine, adenine, guanine, cytosine, and instead of thymine, uracil will be there for the RNA, and for DNA, adenine, guanine, cytosine, thymine. Right? So these are the nitrogenous bases which is required. Second part which is required is sugar. So sugar which is required for the DNA is pentose. Sugar is pentose sugars, 5 carbons, and we also call it ribose, right? So if you see the sugar molecules, so 1, 2, 3, 4 and 5, right? This is the structure of your ribose. So there is a hydroxyl group is there, there is another hydroxyl group, this is the ribose. So if you are synthesizing RNA, then this sugar is okay, right? You can use it. But when we talk about, if you are talking about DNA, so you can see that in DNA का नाम क्या है? Deoxyribose. D is deoxyribose. So this is the ribose. This is not the deoxyribose. तो इसको deoxyribose कैसे होगी ये? This is the deoxyribose where you are removing this oxygen group. So it contains hydrogens, OH, and 1, 2, 3, 4 and 5. This sugar is deoxyribose. This is the ribose, but you have removed the oxygens, right? Deoxy, you remove the oxygen. So it remove the oxygen, it becomes deoxyribose. So for the DNA, this sugar is needed, right? And if you are going to synthesize RNA, then this sugar is okay, right? So sugar molecule which is required is ribose, but for DNA you need deoxyribose, because DNA's name is deoxyribose nucleic acid. And what is RNA? RNA is ribose nucleic acid. So sugar for the RNA is ribose. So this is the ribose, right? And sugar for DNA is deoxyribose. So deoxyribose, you have removed the oxygen, right? So it's a deoxyribose. So sugar for DNA, this one is required, and sugar for RNA, this one is required. And nitrogenous bases, I told you, for RNA you need adenine, guanine, cytosine, uracil for RNA. For DNA you need adenine, guanine, cytosine, thymine. So nitrogenous bases plus sugar molecules, we called it nucleoside. So this combination we call nucleoside. Nucleoside. So what is nucleoside? Nucleoside is equal to nitrogenous bases plus sugar, right? So nucleoside for, if we talk about nucleoside for DNA, so for DNA, nitrogenous bases will be adenine, guanine, cytosine, thymine, and sugar will be deoxy, right? So here it should be hydroxyl group, not oxygen. Here this sugar is required for the DNA. And nucleosides for RNA, nitrogenous bases will be your adenine, guanine, cytosine, uracil, plus sugar molecule will be this one, OH, right? This is the ribose, right? This is the ribose for RNA, because it's a ribose nucleic acid and it's a deoxyribose. So you can see that oxygen is not there. When we talk about RNA, oxygen is not there, and when we talk about RNA, there is a uracil instead of thymine, right? So now it's contained, we called it nucleoside, but as I told you, DNA is not the polymer of nucleoside, it's a polymer of nucleotide. So this nucleoside has to be converted into nucleotide. So how nucleoside will be converted into nucleotide? So when you are adding the phosphate group, so nucleotide is equal to nitrogenous bases plus sugar plus phosphate. So now it's called a nucleotide, and DNA is a polymer of nucleotide. So I am going to discuss how it will be happens. So you know, so for example, this is your sugar molecules, right? This is your adenine, right? So this is your sugar molecules and this is your guanine, right? So if you name this molecule, this is adenosine. Adenine is attached with sugar molecule, we call it adenosine, right? This molecule name is adenosine, right? Sugar molecule attached with guanine, guanosine. Then sugar molecule which is attached with your adenine, guanine, cytosine, then you called it cytidine. When sugar molecule attached with your thymine, then you called it thymidine, right? So these are the nucleoside. Now again, it's not a nucleotide, it's a nucleoside. So to convert into nucleotide, there is a phosphate group will be attached. So this is 1 position, 2nd, 3rd and 4th, 5th. So on the 5th positions, your phosphate will be attached. So if you name these molecules, so now it's become adenosine monophosphate. One molecule is attached, so you will name it adenosine monophosphate. If one molecule is attached here, so you will call it guanosine monophosphate. If one molecule is attached here, cytidine monophosphate, thymidine monophosphate. If two molecule is attached, phosphate molecule, then it will be called adenosine diphosphate, guanosine diphosphate, right? And cytidine diphosphates, thymidine diphosphate. If three molecule attached, then it will become adenosine triphosphate, and that is your ATP, right? Adenosine triphosphate. Here one more molecule is attached, it will become GTP, guanosine triphosphate, like CTP, right, TPP. So this is the nucleotide, right? So nucleotide, now we are talking about this particularly, this is particularly is a your nucleotide, polymer of nucleotide. We talk about DNA, and DNA is a polymer of nucleotide. So we are talking about nucleotide. So you can see that this is your sugar molecules, this is your nitrogenous bases, here is your phosphate molecule is attached, right? Then for example this is your second molecules, right, adenine, guanine, right? So now these are the molecules which we call nucleotides, but to become a polymer they have to be attached with each other, like that, next molecule, and it should become a polymer. तो हमने बात की थी कि डीएनए क्या है, सिंपलर वर्ड्स में डीएनए है पॉलीमर किस चीज़ का, पॉलीमर है न्यूक्लियोटाइड का। और न्यूक्लियोटाइड में क्या क्या है, न्यूक्लियोटाइड में आपका शुगर है, नाइट्रोजीनियस बेस है और फॉस्फेट है। शुगर और नाइट्रोजीनियस बेस बनाता है न्यूक्लियोसाइड, जब फॉस्फेट अटैच हो जाता है दैट इट विल बिकम न्यूक्लियोटाइड। देन दीज मॉलिक्यूल्स शुड बी अटैच विद ईच अदर, ये आपस में अटैच करने चाहिए। सो हाउ इट अटैच, सो दिस इज द फर्स्ट, सेकेंड, थर्ड, फोर्थ एंड फिफ्थ पोजिशंस. So this third positions will be attached with the fifth positions. Here is the linkage. Here linkage will happen, and for this linkage energy is required. So these bond will be broken, and these two phosphate molecule will be removed. That will be called pyrophosphate. And here is in hydroxyl group is there. So this OH and phosphate will be linked together, and that linkage we call phosphodiesterase linkage. Diester linkage. So phosphodiesterase linkage is happening with 3' with 5', right? So 3' there is a hydroxyl group, on 5' there is a phosphate molecules, and this linkage is very strong. Phosphodiesterase linkage is very strong, you can't break it. ये अगर आप टेम्परेचर बढ़ा दोगे या pH चेंज कर दोगे, ये लिंकेज नहीं टूटेगी. To break this linkage, you need specific enzymes, and that enzymes we call DNases. These enzymes can break this phosphodiesterase linkage. So these enzymes is classically we divide into two parts: endonucleases and exonucleases. So there are two types: endonucleases and exonucleases. Endonucleases are the enzymes which can break this linkage anywhere in between, कहीं भी break कर सकता है. But exonucleases are those enzymes which will break at top or the bottom, not in between. या तो कहीं ऊपर break कर सकता है या पीछे break कर सकता है, not in between. That is we called exonucleases. Exo, word exo is there, exo, right? या तो top पे break कर देगा या bottom पे break कर देगा. Endonucleases can break in between as well. तो ये बीच में भी तोड़ सकता है आपका, दैट इज द एंडोन्यूक्लियेजेस. एंड अगेन एज आई सेड, जो आपकी फॉस्फोडाइएस्टरेज लिंकेज है वो कहाँ पे है, 3' और 5'. 3' पे हाइड्रोक्सिल ग्रुप है, 5' पे आपके फॉस्फेट ग्रुप है. तो ये फॉस्फोडाइएस्टरेज लिंकेज बहुत ज़्यादा स्ट्रॉन्ग है. अगर आप टेम्परेचर बढ़ाते हो, पीएच चेंज करते हो, तो ये लिंकेज नहीं तोड़ पाओगे. उसको तोड़ने के लिए यू हैव टू ऐड स्पेसिफिक इंजाइम, और उन इंजाइम का नाम है डीएनएजेस, और वी ऑल्सो कॉल इट न्यूक्लियेजेस. अनदर नेम इज न्यूक्लियेजेस. तो यही इंजाइम्स इस लिंकेज को तोड़ सकते हैं, बाकी इंजाइम नहीं तोड़ सकते, बाकी प्रोसेस नहीं तोड़ सकते. नाउ लाइक दिस इज द पॉलीमर. नाउ एज आई सेड कि डीएनए क्या है, डीएनए इज ए पॉलीमर ऑफ न्यूक्लियोटाइड. नाउ पॉलीमर बन गया, 1, 2 ऐसे तीसरा मॉलिक्यूल, चौथा, पांचवा, लाइक दैट इट विल बिकम पॉलीमर. बट अगेन स्टिल डीएनए इज नॉट कंप्लीटेड बिकॉज डीएनए इज ए डबल स्ट्रैंड, राइट? इट्स ए डबल स्ट्रैंड मॉलिक्यूल्स. सो देयर इज ए कंप्लीमेंट्री मॉलिक्यूल्स, राइट? So there is a sugar molecules, right? Similarly, there is a, for examples, A will bind with thymine and G will bind with C, cytosine. So there is a, now this is a double, these are the complementary with each others. So now A will bind with thymine and they will binding with the double bond, and they will binding with the triple bond, and these bonds are hydrogen bonds. these bonds between adenine and guanine, adenine and thymine, and guanine and cytosine, these bonds are hydrogen bonds. So A is binding with the thymine with the double hydrogen bond, G is binding with cytosine with the triple hydrogen bond, and these hydrogen bonds are strong, not stronger. These hydrogen bonds you can break by increasing the temperature. If you increasing the temperatures, if you changing the pH, so these bond can be broken down. तो जब आपने फॉर एग्जांपल डीएनए लिया, डीएनए को हीट कर दिया, हीट करने पे कौन से बॉन्ड टूटेंगे? फॉस्फोडाइएस्टरेज लिंकेज नहीं टूट पाएगी, बट द बॉन्ड बिटवीन ए एंड टी, जी एंड सी, इट विल बी ब्रोकन डाउन. तो आपके जो डबल स्ट्रैंडेड डीएनए है वो सिंगल स्ट्रैंड में कन्वर्ट हो जाएगा. एंड दैट इज द प्रिंसिपल फॉर द पीसीआर, राइट. आई होप एवरीवन ऑफ यू नोज अबाउट पीसीआर. कोविड टाइम में बहुत सारा आरटी-पीसीआर करवाया गया है. पॉलीमरेज चेन रिएक्शंस, दिस इज द टेक्निक बाय विच यू आर एम्पलीफाइंग द डीएनए. एम्पलीफाई कैसे करते हो डीएनए को? पहले आप डीएनए की स्ट्रैंड्स को सिंगल करते हो, देन यू आर एडिंग प्राइमर, न्यूक्लियोटाइड, तो उसके आपकी कॉपीज बनती हैं. तो व्हेन यू आर, इफ़ यू सी द पीसीआर में पहला स्टेप है डीनेचुरेशन, एंड इन डीनेचुरेशन यू आर इंक्रीजिंग द टेम्परेचर. 92 डिग्री सेल्सियस तक आप टेम्परेचर ले जाते हो. तो जब आपका टेम्परेचर हाई हुआ तो दिस स्ट्रैंड विल बी ब्रोकन डाउन, these hydrogen bonds will be broken down, right. So this is the principle. So these hydrogen bonds will be broken down, and again जो A है that is binding with the T through the double hydrogen bond, and G is binding with the cytosine with the triple hydrogen bonds. So if you have a two strands, for examples you have a two DNA strands, right, and you know this DNA strands GC content is more, and this DNA strand has GC content is less. So which strand you can break easily? You can break easily these strands because GC content is less. Why it is so? Because G is triple hydrogen bond and A is binding with double hydrogen bonds, right. So that is the difference, you know, because GC content is less, so that strand can be broken down at lower temperature as compared to this strands where GC content is more. For the strand which have a more GC content, you need more temperature because this has a more triple bonds, right. GC content is more, right. So that is about your DNA. But again for the DNA, quickly if I tell, it's a polymer of nucleotide, polymer of nucleotide. So now question is, what is nucleotide? Nucleotide is nitrogenous bases, nitrogenous bases plus sugar, and sugar will be pentose plus phosphate, that is your nucleotide, right, right. So nitrogenous bases is your purine and pyrimidine. In purine there is adenine, guanine, and in pyrimidine cytosine, thymine, and instead of thymine if uracil is there, that is for RNA. Sugar is your pentose sugars, we are also called ribose, and ribose sugars you are using for RNA, and for DNA you have to remove the oxygen, that's why we called deoxyribose nucleic acids, and phosphate will be same. And these linkage, phosphodiesterase linkage will be done with 5' to 3', right, 3' to 5', 3', like that. This is linkage is very strong, that we call phosphodiesterase linkage. You can't break it, you have to add the specific enzymes like DNases, nucleases, exonucleases or endonucleases to break this linkage. But complementary, these adenine will be with thymine and G is binding with thymine, and these are the hydrogen bond which can be break easily by increasing temperature as well. Classical example is PCR, right. And with respect to DNA, every one of you might know that DNA is expressed into RNA, right, and RNA is expressed in the proteins, right. So we call nowadays, you might heard about proteomics, some is talking about transcriptomics, some are talking about metabolomics, right. So DNA, RNA to protein, and proteins are of two types, structural proteins and functional proteins, structural and functional proteins. So depending upon structural proteins, आपके नाक का साइज कितना होगा, आपके आँख का क्या कलर होगा, that is your structural protein. How it function, that is your functional proteins, right. So DNA to RNA we call it, this step is transcriptions, and RNA to proteins we called it translations, right? And this is we also called a central dogma of life. So this is about your DNAs, and of course there is a Chargaff rule is there, right? Chargaff rule is A will T and G will complementary to C. That is your Chargaff rule, right? Thank you. This is all about your DNA, right? This is about your DNAs, about the cell components. Thank you.